Thank you very much, David, for a very nice introduction. So, thank you very much for all coming to this early morning session. And I also would like to thank organizers for giving me this opportunity to talk in this really interesting first INCF Congress. So today I'd like to cover rather wide areas of humanoid robotics inspired by neuroscience and cerebral computational model. And third is a systems biology model of synaptic plasticity at single spine of cerebral Purkinje-cell. And finally, I would like to propose a manipulative neuroscience based on some technology from brain-machine interface. So as a self-introduction video, I would like to show a Discovery Channel broadcasted in the US several years ago.
To get a robot worthy of our sci-fi fantasies, you need to give it intelligence. And that's what neurophysicist Mitsuo Kawato and Stefan Schall are up to.
This is how time will be called.
They run the Kawato Dynamic Brain Project at the research center near Kyoto, Japan. Their star pupil? A robot named DB, short for Dynamic Brain. They've designed a brain program for DB that allows him to learn motor skills. So far, the two scientists have taught DB to play a drum, balance a pole on his finger, bounce a ball on a racket, and cut some pretty groovy dance moves. The amazing thing is, DB doesn't pick up these skills by being programmed with them. Instead, he observes with his video camera eyes, and then tries and tries to mimic behavior till he gets it right.
If robot can imitate human motor skills or human motor tasks, that is fascinating because we do not need to write programs by ourselves. Robot can learn by himself, by imitation.
Okay, we've got a robot smart enough to ape our movements. Way cool.
So this is a robot named DB after Dynamic Brain Project, which we did until 2001. Now we've been expanding this research into new types of human robots. Here I show you two new robots, and this one is called CBI, Computational Brain Interface, which has been developed in collaboration with Professor Chris Atkinson at CMU and Circus Research Company located in the University of Utah. So this robot is even closer to human in its speed and power than DB, and this is fully autonomous because it has computer and electricity and oil pump on all his back. And it was supposed to be 160 centimeter and 60 kilogram, but with a power pack, it is now 85 kilogram. And the most important feature of this is it's mechanically compliant because it uses hydraulic actuators and does not use electrical motors, so it does not need reduction gears at the joint, which makes a human-like compliance to his body. And this robot can do several things like baseball batting or posture balance under very strong and unexpected forces. But one of the most exciting achievement which we had with Professor Miguel Nicolelis at Duke University is controlling this robot by a monkey brain situated in the east coast of the United States. So we established bidirectional internet connections between monkey brain in Duke University and CBI in ATR Kyoto Japan. I'm sure that Miguel will talk about this on Tuesday. So I will show you just a few clips. This is a biped locomotion based on central pattern generator of CBI, and this is experimental video, which shows a real-time bidirectional connection between humanoid robots and monkey locomotion. So we utilize technique of decoding locomotion pattern by Miguel's lab and send it this information from Duke University to Kyoto by internet, and the robot moves according to this desired movement pattern by monkey, and then video is shoot for this locomotion, and it is sent back to Duke University by the method of video streaming. And the monkey is excited to see robot walking by his command. Because we have only one over several sounds time delay for this bidirectional transmission, information transmission, monkeys seem to fully realize that the robot is under his control. And we have about 80 researchers, secretaries, and students in a group. This is a photograph from the last year Hawaiian-style barbecue party in the summer. And ATR Computational Neuroscience Laboratory is funded by several sources. One, robotics part is funded by JST ICOP, a computational brain project with Carnegie Mellon University, with main collaborator Chris Atkinson. Now, Advanced Institute of Science and Technology have contracted with us, so we have about 10 nice students, and the student number is something like 25 to 30, including non-Japanese students. And National Institute of Communication Technology is recently increasing source of funds. And we are in collaboration with Honda Automobile Company developing reinforcement learning for robots, as well as brain-machine interface control of human robots, which I'm going to tell you. Now, where is ATL, this ATL located? ATL is located in the south border of Kyoto Prefecture, and which is very conveniently located between two highest sightseeing cities in Japan, Kyoto City, which is full of beautiful Japanese gardens within shrines and temples. And we have Nara, which is only 25 minutes drive from ATL, and Nara is famous with big Buddha and also very tame but wild deers. Although these deers are quite tame, they could destroy your beautiful gardens or eat vegetables from your farms. So Japanese invented a tool to scare deers to protect their gardens. This is called Shishi Odoshi in Japanese. If I translate it directly into English, it's deer scaring. Deer scaring is a bamboo tube which is pivot and can rotate around this pivot. And one end of this bamboo tube is open, and water flow can flow into this half side of the bamboo tube. And if this side becomes heavier, then it rotates around the pivot, and all water going out, and then it makes rotation back, and it makes a large noise to scare deer. So I assume that most of you are familiar with this Shishi Odoshi. So let me show you American Japanese gang movie called Kill Bill. In one scene of this Kill Bill, Shishi Odoshi makes a very important role. Here you can see two non-Japanese yakuza fighting with each other, but on this side you could see this deer scaling, water flow into this and makes this noise, right? So today one of my take-home message to you is that cerebellar Purkinje-cell long-term depletion is like Shishi Odoshi. So here we have a dendritic spines and dendritic shafts of single Purkinje cell. The Purkinje cell received two major excitatory synaptic inputs. One is climbing fiber inputs. Only one axon of this climbing fiber innervates a dendritic shaft, and this is very strong excitatory input. In contrast, we have hundreds of thousands of parallel fiber inputs to Purkinje-cell, and all of the parallel fiber inputs are made on head of dendritic spines. And it is postulated that climbing fiber is carrying error signal of some motor control outcome. So, in correspondence to this, if parallel fiber input is excited preceding about 100 milliseconds to resulting climbing fiber inputs, then calcium concentration within this dendritic spine increases. And then we have some signaling cascade, and the AMPA receptors, which are the major synaptic receptors at this parallel fiber Purkinje cell, number decreases. So this is a known signal transduction pathway for single long-term depression, single spine long-term depression. And I try to persuade that this is implemented by a MAP kinase positive feedback loop, which has bistable dynamics, so which corresponds to this bamboo tube. And water flow is actually replaced by calcium. Large calcium impulse is generated by combining climbing fiber and parallel fiber inputs. Then, if this crosses the threshold, this MAP kinase positive feedback loop goes to an upper, higher equilibrium state, which is stable. And then PKC, activated PKC, phosphorylates AMPA receptor, and phosphorylated AMPA receptor tend to be exocytotic. That is, these AMPA receptors are detached from the membrane. So synaptic efficacy goes down. So let me give you several backgrounds for this main story. As you may know, neural circuit of the cerebellar cortex is crystal-like, which is very uniform and beautiful. The only output neurons of the cerebellar cortex is Purkinje cells, whose axon goes down to cerebellar deep nuclei. And as I said, a Purkinje cell receives two major excitatory synaptic input. One is climbing fiber, and the other are numerous parallel fibers. So David Marr, James Albers, and Masao Ito, almost 40 years ago, based mainly on this anatomical arrangement, postulated that climbing fiber inputs act as a teacher or error signal, and parallel fiber and Purkinje cell synapse undergoes some synaptic efficacy change when climbing fiber and parallel fiber are almost simultaneously excited. Then we postulated that resulting synaptic plasticity can make internal models of some external world within this cerebellar cortex. Let me very briefly explain our cerebellar internal model theory. It is known that the cerebellum consists of many micromodules called microzones, maybe 50,000 to 100,000, which perform different input and output transformations. Synaptic weights, that is, parallel fiber to Purkinje cell synaptic weights can change, and different transformations can be learned individually in these different modules. And supervised learning takes place while guided by an error signal carried by climbing fiber inputs. As a result, different modules can acquire internal models of controlled object, tools, and other brains, and so on. Let me show you some robotics demonstration of this. So here is Professor Stefan Schall, who has been my main collaborator in robotics research. He was a group leader of my LADA project from 1996 to 2001. He's now a professor at the University of Southern California. So this is pole balancing. We didn't do any hack. This is really free. We didn't use any paste or joint.
We applied the developed statistical learning method to various tasks in order to verify its effectiveness. Visual motor learning is one possible task. In this task, the robot should keep tracking a ball with its hand, which requires the learning of self-dynamics information, as well as coordination between the target and the hand. After the robot finishes learning the task, if we put a weight in the robot's hand, the robot can no longer track the ball. However, it continues to learn its new dynamics, and finally recovers and achieves good visual motor coordination. The robot can update its internal model when it encounters a change in the model.
So in the former half of this pole balancing, so-called forward internal model of the pole was used. But in the latter half of visually guided reaching, we used inverse kinematics and inverse dynamics model of the arm for reaching. So computationally, we proposed some useful and efficient algorithm called feedback error learning, how to learn a cerebral internal inverse dynamics model or inverse kinematics model. Then we were quite fortunate to collaborate with neurophysiologist Kenji Kawato and others to examine the prediction of inverse dynamics model. In the case of eye movement, eyes in one dimension, inverse dynamics model became very simple. It's just a linear summation of acceleration of eye rotation, velocity, and position, and this simple linear equation can explain quite different complicated waveforms of single Purkinje cell, which are shown by black dots by a theory, a red line, for more than 70% of the neuron with squared correlation coefficient larger than 0.7. And now we accumulated many experimental supports to cerebellar internal models, and we are quite certain that it is a correct theory for ventral power focus of the cerebellum during ocular following responses. But this is only 1/1000 of total cerebellum. Other evidences accumulating from different labs, for example, climbing fiber inputs encode motor command errors and not sensory error. This is the essential, most essential assumption in our theory, and this was validated by this work. And also, strong climbing fiber inputs in delta 2 knockout mouse induce very poor control. This is also in agreement with our prediction. And the chaotic resonance induced by electrical synapses in inferior olive is efficient learning with low firing rates. Now we examine the data from inferior olive obtained by Eric Lang and Rudolf Linus and found that these data support our hypothesis. But we still have controversy. For example, for arm movements, Kenji Yamamoto and myself published Journal of Neurophysiology saying that cerebral Purkinje cell neurons encode different dynamics while monkeys are manipulating robotic manipulum with artificial force field. But in almost the same experimental paradigm, Tim Ebner's group published a Nature Neuroscience paper in 2006 proposing the opposite conclusion. So I will come back to this difficulty at the end, towards the end of my talk. Now, yesterday, Mary Kennedy scared us with very complicated signal transduction circuit for cellular cortex and hippocampus, synaptic plasticity, LTD and LTP. And I am proud that we have similar thing for cerebellar long-term depletion. Probably cerebellar long-term depletion is better studied. But even if you look at this signal transduction summary for hours and hours, you cannot answer the following questions. So the basic questions for systems biology model of cerebellar LTD are the following. So we would like to understand memory. What is memory? It's a temporal transformation from short-term events of 100 milliseconds, which consists of only several parallel fiber spikes and single climbing fiber spikes, which induce long-term depletion and which leads into long-term events of several years. For example, if we once learned to ride on bicycle, even if we do not do that exercise for 20 years, but still we can instantaneously decode that kind of memory. And people believe that the cerebellum is a site of this kind of long-term memory. So how can this happen? And the stochasticity due to small number of molecules and thermal noise, and this is a big question. The second is mechanism of spike timing dependent plasticity, which you also had from Mary Kennedy's yesterday's talk. Because, as I already explained, because climbing fiber carries resulting error signal of motor control or cognitive control, long-term depletion is optimally induced when climbing fiber is delayed with respect to prior fiber for 100 to 100 milliseconds. How molecular machinery can generate this temporal delay? And in many different laboratories, several non-associative LTD data was obtained in different experimental paradigms. For example, only calcium injection or only IP3 or only bundle stimulation of parallel fiber can induce LTD. Then it is in odd with associative nature of supervised learning. In supervised learning, you need a combination of climbing fiber and parallel fiber. So with these confusing experimental data, serious doubts and criticism on LTD as a cellular basis of cerebellar supervised learning, or in more general, cerebellar learning, were cast 20 years ago to quite recently. So we would like to explain all these with a single systems biology model of long-term depletion within a single dendritic spine of Purkinje cell. So this is a summary figure of two kinds of model of cerebellar LTD. One essential feature of this, first of all, I need to explain. A climbing fiber and parallel fiber induce different effects on single dendritic spine. Parallel fiber can induce NO, nitric oxide, and which will finally influence the activity of this positive feedback loop of comprising of PKC, LAF, MEK, MAP kinase, PLA2, AA, arachidonic acid back to PKC. And parallel fiber also have glutamate, and glutamate induce depolarization through AMPA receptor. So this is a mainstream synaptic input. But at the same time, glutamate binds to mGlual receptor, and G proteins generated, and PLC generated, and IP3 and diacylglycerol is generated. So we have, for parallel fiber, we have these three pathways of signal transduction. On the other hand, climbing fiber has a relatively simple depolarization, which induces opening of voltage-gated calcium channel on the dendritic spine. So calcium goes into this. So, Shinya Kuroda's main proposal is that IP3 receptor on the calcium store within dendritic spine is a kind of coincidence detector of parallel fiber input represented by IP3 increase and climbing fiber input represented by calcium increase. And this detects a coincidence of these two events, and if these two are simultaneous, then large calcium-induced calcium increase is generated. And this calcium triggers this MAP kinase positive feedback loop, which has two stable equilibrium. And at a high stable equilibrium, PKC is activated and it phosphorylates AMPA receptor, and then AMPA receptor is detached from the membrane. So this is the main story. Okay. First, let me explain how spike timing dependent plasticity or temporal window of LTD is generated. The essence is that although the depolarization and calcium inflow from the outside of the cell is a very fast, immediate event, but the mGlual pathway, metabotropic glutamate receptor pathway to generate IP3 takes time, something like 100 to 300 milliseconds, quite slow process. So only when parallel fiber precedes climbing fiber, say 100 milliseconds, then 100 milliseconds after IP3's concentration is maximum, Then if climbing fiber comes in, you have large calcium inflow, which is simultaneous with IP3 increase, then IP3 receptor is open. So a delay in slow prior fiber metabotropic pathway compared with fast climbing fiber electrical pathway as a mechanism for timing detection. And we have fast positive feedback loop, that is larger calcium concentration in the cytosol tends to open IP3 receptor more, so more calcium goes out, so we have fast positive feedback loop. But at the same time, we have slow negative feedback loop for IP3 receptor, so too high calcium concentration shuts down IP3 receptor. So we have this kind of calcium spike with 200 millisecond width triggered by lower threshold because of higher IP3 concentration. And with this, this is ordinary differential equation type model. So we have only 3 compartments: postsynaptic density, cytosol, and calcium store. And we have well-mixed assumption. And we also have large number of molecules assumption because we are dealing with ordinary differential equation. Towards the end of the system biology model of LTD, I will go to more realistic stochastic simulation based on Gillespie's algorithm. Okay, with this, we can explain temporal window of long-term depression. That is, if climbing fiber precedes parallel fiber too early, nothing happens. But if climbing fiber is almost simultaneous with parallel fiber excitation, or climbing fiber is about 200 millisecond delayed, you have large calcium increase within a single dendritic spine, which is almost 10 micromolar. But if climbing fiber is too much delayed with respect to parallel fiber, calcium increase is small. Right. So we at least explain the spike timing dependent plasticity mechanism. Then how about memory? The essence of this model is bistable dynamics of MAP kinase positive feedback loop. This was first proposed by Upibala as a generic model for hippocampal or cerebral cortex LTD, LTP. And then Shinya Kuroda adapted this model to Purkinje-cell LTD. Interestingly, we have strong supports to cerebellar story, but not so in cerebral cortex or hippocampus when I heard yesterday's talk. So anyway, we have 2 stable nodes separated by a saddle, and the stable manifold of this saddle is the separatrix of GS2. So if you give strong and long enough calcium input, the state can jump across this separatrix and can go to this state, which is LTD state. If I am allowed to draw this with some kind of pseudo energy, you have double well. And in order to induce LTD, you need to climb up this hill and go into this direction. This is done by calcium inflow. We are quite fortunate that experimenter Professor George Augustine and Keiko Tanaka tested several essential predictions of this model under the Human Frontier Science Program collaboration led by Eric Deschatter. So they did calcium, this is George Augustine, this is Keiko Tanaka and her husband. And they did calcium photoencaging control of 30 dendritic spines. And at the same time, they measured calcium by confocal microscope. So the experimental setup seems quite complicated. And you have Purkinje cell, you have patch clamp at the soma, and they injected caged calcium as well as calcium dye. They have objective, and you have confocal microscope imaging, as well as by giving light source, you could control the concentration of calcium within this area. And you have parallel fiber inputs to this area. They put the electrode, stimulating electrode, quite close to this optical imaging and optical control. So in collaboration with them, we published a paper in 2007 in Neuron. And this is the dendritic shaft. And it is difficult to see, but we have small dendritic spines. And this shows the spatial extent of calcium uncaging. If you plot the calcium concentration as a function of distance within 10 micromolar, 10 micrometer, you have this kind of distribution. Regarding temporal resolution, if you give laser pulse, you have highest calcium concentration within 100 or even less millisecond. So this is collaboration with Keiko Tanaka, George Augustine, and others. And this study showed that LTD is most probably all or nothing behavior at single synapse. And we have leaky integration of calcium by MAP kinase positive feedback loop. And we have resulting by stability. Let me introduce their experimental results a little bit. So they could measure synaptic efficacy as parallel fiber EPSC at the voltage clamped soma. So if you do not do anything, then synaptic efficacy remains quite stable for 10 minutes, 20 minutes, but then they give a flash for photo-uncaging of calcium. Then for the flash one, calcium concentration is something like 0.5 micromolar, but the synaptic efficacy remains the same. For the second flash, it's 1 micromolar, but still synaptic efficacy is constant. But for the flash 3, which is 1.5 micromolar at the highest, then synaptic efficacy dramatically decreased with a time course of something like 10, 20, 30 minutes. Okay, so this is very typical long-term depletion in slice preparation. Then Keiko Tanaka and George systematically changed the calcium amplitude and the duration. And they plotted long-term depletion expressed as a function of highest calcium concentration as well as the duration of the concentration. You could notice that for longer 30-second duration with 1.5 micromolar calcium, you could have already maximum long-term depletion. But for very brief pulse, 0.5 second, you need to have 6 micromolar or 5 micromolar concentration to attain the maximum long-term depletion. So you have this sigmoid curve with quite high Hill coefficients in this system. Now, we can well explain this quantitative data by Leaky integrate and Depress model. So this is very similar to Leaky integrate and fire model. In the case of neuron model, you are integrating electrical current to produce voltage. In this case, you are integrating calcium input with some leak to give the amount of long-term depletion. So X is long-term depletion, which decays, and calcium is input. And with this transformation, although the data are scattered depending on the duration of calcium spike duration, but if we transform this, all the data is lying on a single theoretical curve. So it seems that you have leaky integrator of calcium. Also, in simulation, you could easily cut some part of the MAP kinase positive feedback loop. Then bistability is gone, nonlinearity is gone. And in that case, plots of LTD as a function of calcium or plots of LTD as an integral of calcium have very different pictures from the previous control condition. Right? In the control condition, all the curves seem to go to the same plateau level. But in the case of cut MAP kinase positive feedback loop, G's are different and curves are crossing with each other, which did not happen for control. And experimental reproduction of these is quite remarkable. And furthermore, Keiko Tanaka and George Augustine published another neuron paper this year utilizing molecular knockdown method. So they show that MAP kinase and PKC are exciting with each other to form a positive feedback loop, which is essential for induction of long-term depression. So positive feedback loop summary is as follows. It seems that each parafiber Purkinje-cell synapse on the spine, single spine, undergoes LTD in all or none manner. We have several pieces of evidence. One is cooperativity, that is large Hill coefficient in calcium versus LTD dose response curve. And the second is constant maximum LTD irrespective of duration of calcium increase. And the third is reproduction of experimental data by bistable dynamics. Bistable dynamics is all or none. MAP kinase positive feedback loop provides the second threshold dynamics for LTD. The first dynamics is fast calcium excitable dynamics determines the temporal window of spike timing dependent plasticity. And then we have this second MAP kinase bistable dynamics. And between these two excitable and bistable dynamics, we have Leaky integrator between two nonlinear dynamics. So we are happy to obtain several experimental supports to this Shishi Odoshi de-scaling view of all or nothing long-term depression in cerebral Purkinje cell. And is the story final? No, not at all. First of all, I would like to explain the difference of this cerebral LTD versus cerebral cortex or hippocampus LTD or LTP. So I'm very sorry that in even famous computational neuroscience textbooks, Purkinje-cell LTD is described as anti-Hebbian. But Purkinje-cell LTD is by no means anti-Hebbian. First of all, we do not have backpropagation of somatic potentials because of too much dendritic branching. So if somatic action potential does not backpropagate, there's no way of implementing Hebbian learning. And furthermore, there's no active NMDA receptor in hippocampus and cerebral cortex. NMDA is postulated to be the key of spike timing dependent plasticity, but we don't have that machinery in cerebellar Purkinje cell. And spike timing dependent plasticity temporal window is 100 millisecond order in cerebellar Purkinje cell, probably in order to dealing with motor control issue. But as you had yesterday in hippocampus or in cerebral cortex, it's 10 millisecond order or even less. So we have large difference in temporal window. So with this, I conclude that David Ma, James Albers, and Masao Ito was correct. Even from the systems biology modeling of cerebellar long-term depression, which is so different from cerebral cortex or hippocampus. So calcium, which is carried by climbing fiber input, is error signal. And synaptic plasticity doesn't care whether cell is firing or not. So it's not Hebbian at all. It is supervised learning controlled by climbing fiber inputs. So I think this kind of small-scale systems biology modeling can somehow relax controversy at the system level. But we need to think about plasticity and stability dilemma more. In a sense, all synaptic plasticity needs to satisfy this plasticity stability dilemma. That is, synapse should be sensitive enough to a few pulses occurring within 100 milliseconds. But somehow, memory must be retained for years. How can it happen with only 20 to 40 ampere receptors in single dendritic spine of Purkinje cell? This is an estimate made by Japanese recently. If you have only 20-40 ampere receptor, you have stochasticity. That means even if you are in this LTD state, because of thermal noise, because of accidental collision of a small number of molecules, you may come back to this basal state. The memory is lost. How can we avoid this? We've been working on nested or cascaded positive feedback loop under stochastic situation. So Hideaki Ogasawa is my main collaborator on this issue. So we made a very simple differential equation which explains the essential feature of positive feedback loop. Here, x excites y, y excites x, and if we introduce some kind of nonlinearity, then phase plane analysis shows that we have two stable nodes here, one saddle here. So with the input stimulus, this state could go to this upper state, which corresponds to long-term depression. If you give very brief input, then the system goes to high state. If you give, we have here time constant, which determines time constant of this positive feedback loop, irrespective of differences of time constant. Stimulus always makes the state from here to here. So for single deterministic positive feedback loop, we could see threshold phenomena. If you give weak stimulus, y goes a little bit higher, but it is smaller than the threshold, so it goes down to the basal state. But if you give strong enough, that is, amplitude is large or duration is large, then the state goes to upper stable state, so it stays there indefinitely. If we believe in deterministic situation. But in stochastic simulation, we have here fast positive feedback loop and slow positive feedback loop, and the output of this fast positive feedback loop y goes into z as the input. Here, situation is much more complicated. Here I plot y, the output of the fast positive feedback loop. W is the output of the slow positive feedback loop. Now, we use Gillespie algorithm, so rigorous stochastic simulation solver. Each trial is different. In this case, fast goes to the stable, high stable equilibrium, but because of noise, it goes down to the basal state. But if the time at the higher state is long enough, this gives enough input to the slower dynamics, so it tends to stay at the high equilibrium. Because the time constant of this slow positive feedback loop is much larger than this, memory is lost here very quickly, but the memory can be retained for a longer time. And so we are now proposing conceptually this kind of cascade of excitable and stable dynamics models with which we could have high sensitivity to small number of pulses as well as long-term memory, even under the stochastic situation. So from this study, which was quite new to me, I saw direct manipulation of information is really important. So information can be directly observed and controlled in this neuron study. But information cannot be directly observed or controlled in usual system neuroscience. In system neuroscience, what you usually do is correlation analysis. So we believe manipulative system neuroscience is really necessary. Well, we believe necessity to link theory and experiments beyond mere temporal correlation of hypothetical theoretical variables with neural firings or brain activation. So decoding of neural information by brain network interface and its feedback to brain seems to be essential. So with this goal, we've been trying to combine fMRI and MEG and news with EEG to obtain high spatial temporal resolution. For example, we could reproduce reliably four-quadrant activation of the primary visual cortex by combining MEG and fMRI under variational Bayesian technique. We utilize fMRI data as a quite flexible prior information to solve inverse problem of Meng. And with this technique, we could now reconstruct two-dimensional wrist motion from fMRI and Meng. So we do 8-direction movement, something like this, and we utilize fMRI as a prior information and solve Meng inverse problem with this prior. And then within the brain, we have something like 4,000 electrical sources. And from these, we use some kind of a Bayesian technique of linear regression, sparse linear regression. And then the result is quite remarkable with our best subject and best trials. Important thing is that we didn't use any, training session for this. This is just we took movements and brain activity, and from brain activity we could reconstruct list movement for test data. So with this, I believe that we could probably construct so-called brain network interface, which is similar to brain-machine interface or brain-computer interface, but a little bit different. So we try to combine fMRI-Meng or functional news and EEG, first solve inverse problem before reconstruction for BNI, avoiding artifact and noises by constraining currents used for reconstruction only in physiological reasonable brain lesion, and high performance because of efficient feature extraction in brain space. So power estimation of different kinds of information for hierarchical BNI based on computational model of sensory motor control seems to be a future of brain-machine interface. So in collaboration with Shimazu, we already established this combined headset for our functional news and EEG, 60 channels for functional news and 90 channels for EEG. So Japan, our Japanese government, next Ministry of Education, Science and Sports and Culture has just launched $20 million per year for 5 years project on brain-machine interface and genetic control of primates this year. And I am director of the BMI part. So within our group, we have about 30 different research laboratories from universities and national institute. And we have mainly 4 projects. One is EcoG project, and one is neurorehabilitation. The other is multi-channel electrode array and database, and non-invasive BMI, which is combined fNIRS and EEG. I do not have time to explain all of these, but one important aspect of this is to build database for BMI, including EcoG, EEG, fNIRS, fMRI, and multi-electrode channel. And we would like to open this to the world. So in that sense, by constructing BMI-specific database, we would like to contribute to INCF. Thank you very much for your attention.
Thank you very much for a wonderful array of talks about all in 50 minutes about many subjects. There'll be lots of questions, I'm sure, but we don't have all that time left. But are there a couple of questions? Are there a couple of questions that you would like to ask? As I said, maybe my microphone wasn't switched on there, but an amazing talk, lots of things to talk about. A couple of questions. Mark?
Yeah, considering your biostability of your synapse, would it also be the case then that once you LTD a synapse and you wait longer and try again to LTD, it will not be LTD again? Is that the case? Experiment?
Yes. prediction is so, but I don't think researchers has not done that from obvious technical reason. You need to keep, you can keep your preparation, slice preparation only 90 minutes at most. Yes.
Do you see any role in this feedback from Purkinje cells to backward to climbing fibers, which might mark this described, and it could be just general positive feedback between Purkinje cells and climbing fibers. How it works? Do you take it into account?
Are you talking about the Purkinje-cell to deep cerebellar nuclei and inferior only back to climbing fiber? Yes, I think that's really important. And as Rudolf Linus and many others have established, probably by activation of Purkinje-cell, you have inhibition of deep cerebellar nuclei, which reduces GABAergic input to glomeruli of connecting, inferior olive neurons. And this decrease will increase the synaptic coupling between inferior olive neurons. So you are going to have more rhythmic and more synchronized climbing fiber inputs. And I believe that this should be very important at the beginning of learning. Probably it will accelerate long-term depression and cerebellar learning. Actually, we are now collaborating with Eric Lang, who is one of the student of Rudolf Linus, to develop this kind of theory.
I have two questions, if possible, otherwise only one. Okay. One is, you say that timing the cerebellum is probably mediated by this kind of phenomenon. But we know that, for instance, in classical conditioning, cerebellum is involved, and you can learn time scale of seconds. So one question is, how do you explain this? And the second one is, other people, Smoke, for instance, have talked about plasticity in the deep nucleus, responsible for this kind of very long-term memory. So again, how does fit within your framework?
Yeah, that's a very important question. Actually, I think our theory predicts different temporal window between IP3 receptor activation and parallel fiber, depending on what kind of motor behaviors are regulated by that part of the cerebellum. So by changing kind of chemical plasticity within a signal transduction pathway, I could imagine that you could deal with from 50 milliseconds to something like 500 millisecond temporal window. But from eye-blink conditioning type study, we well know that a much larger temporal delay is also allowed. In that case, I believe that the hippocampus or interpositus or other brain areas are coming in. But within, I would say, 500 millisecond to 50 millisecond temporal window, probably this chemical transduction machinery can deal with by changing its pathway a little bit. Did I answer your question? A deep nucleus, yes. It's, in a sense, outside of my scope. Okay.
I'm sure there are many more questions. I think we'll have to leave it there because we've run out of time and we need to go on to the next session. But firstly, can we thank Professor Kawato very much for his amazing talk. Thank you.
Thank you very much. Arigatou gozaimasu. Thank you.
So we now go on to the second workshop of the Congress, and I'll hand over to my colleague Robert Williams, who's going to introduce the speakers.